Hey everybody, this is Katie, one of the biochem TAs, and in this video we're going to be talking about hearing. At the beginning of each video part, I'm going to be asking a question, and hopefully by the end of the video you'll be able to answer this question. So for this video, the question is, how is a sound wave processed into a signal the brain can interpret? So to begin with, when a sound is created, it propagates through the air as a wave and it gets funneled into the ear. It hits the eardrum and these vibrations are transferred into mechanical motion through these bones, collectively called the ossicles. And these bones make contact with a structure called the cochlea. This structure is a fluid-filled compartment that contains hair cells, which are the sensory cells in the system. So what happens is when mechanical motion is transferred to the cochlea, it has fluid inside of it and it propagates those vibrations in the fluid. So taking a closer look of the important cells that are inside the cochlea, these right here are the hair cells, and the fluid runs over the top, and it makes contact with these structures called the stereocilia. So we're going to take a closer look at that here. Like I said, the hair cells are the sensory cell in the system. So what you have, just quickly to go over the individual structures, you have the largest projection is called the kinocilium, and then you have the shorter projections. You have the shorter projections, which are called stereocilia, and this overall structure right here is the hair cell, and then this is a auditory neuron. And so what you have is the fluid is when the vibrations transfer to the fluid, you have the fluid pushes over on the stereocilia, and when that happens, the stereocilia bend, and what takes place is the cell begins to depolarize, so it becomes more positive because ions flow into the cell, causing it to depolarize. And then what happens is that signal is then transmitted to the next neuron, and then the signal is transmitted ultimately to the brain. So this shows a graph of this process where the displacement towards the tallest part, where it's referring to the kinocilium, in nanometers, and then this acts as the depolarization, so the cell becoming positive. So what happens is when there's displacement towards the tallest part, the kinocilia, then what happens is the cell depolarizes, as you can see from this graph. And so you can tell that further displacement causes a higher level of depolarization. And so ultimately what you have is the mechanical the mechanical stimulus gets converted into an electrical signal that then gets sent to the brain to be interpreted. So the next question I would like to address is, why is sound processed so quickly? So I mean this in terms of the transduction process, where you are taking a mechanical stimulus going to an electrical signal inside the hair cell. So this process is uniquely fast in comparison to the other sensory transduction processes. So the reason why that is because when you generate a sound wave comes in to one ear, and in order to be able to tell the direction at which the sound is coming from, you need to be able to detect a time delay, and that is the time it takes for a sound to hit one ear, to arrive at one ear, and then arrive at the other. So when you have that sound come into one ear, it needs to be fast enough so that it can start sending the signal up to the brain, at least start the signal, not necessarily all the way, but it needs to at least start the signal so that by the time the wave gets to the other ear, that the other ear, the signal is further ahead than in this ear. And so in this way, you have two parameters. You have the speed of sound that you're considering, and you have the distance between your ears. And with these two constraints, in order to have a time delay, in order to be able to detect where something is in space, to tell a direction, you have to be able to achieve time delays that are 0.7 milliseconds. So this process that we'll talk about soon, how exactly does it do it so quick in comparison to other sensory transduction pathways, hearing is able to achieve very fast time intervals. In this video, I'd like to talk about the cellular events that take place in the hearing sense. So the question that I have is, what cellular mechanism allows a mechanical stimulus to be transduced so quickly? So I want to begin by highlighting more structures that I didn't draw in the first video. And a really important player in this is going to be this structure right here, shown with the purple coil, called a tip link. And these link together adjacent stereocilia. And the other important part of this is what is attached to the tip link, which is this channel. So the tip links and the channels are coupled meaning they act together. Um, and so we're going to take a closer look at that down below here, but just to remind you, when fluid in the cochlea runs over the stereocilia, what happens is you have this deflection towards the kinocilia of the stereocilia. So they go towards their higher end. And when they do that, what happens is depolarization. And so taking a closer look at the tip links, and so these two pictures correspond to each other. So when you have the stereocilia standing straight up, what you have is there's not a lot of tension in this coil-like structure, which is the tip link. But when the stereocilia start to bend, you start to generate more tension in the tip link, and that causes the channel to open up right here. So this channel right here is opened up, and so what happens is that at this very top you have potassium and calcium rush into the cell by these channels. So what happens is they go in through all of these channels, calcium and potassium, and so they begin to depolarize the cell. And when that happens, it encourages the binding of these vesicles. It causes the binding of these vesicles that are filled with neurotransmitters to fuse to the cell membrane and be released into this place called the synapse. And then the signal, these chemical signals are then taken up by the next neuron in the pathway, and then this signal is carried up to the brain, which can then decipher the meaning. So this is this is a hair cell. This is the image of just the top portion. So you're looking right here at this cone. This is the stereocilia. And down here, you're actually, right here is the tip link. And in your book, they have this figure right here. And I want to just talk about it a little bit because in my picture, I drew the favored forms of this. But in actuality, you have this kind of equilibrium that exists between, I drew the two blue circled ones. That's what I drew here. But like I said, you have an equilibrium between these two different states. And the truth is that about 15% of the channels are open at any given time, regardless, even if they're standing straight up. But I just wanted to highlight this in case you go to your book and you look at what it says here. So the point here is that most of the time, if they're going to be bent in this direction right here, most of the time, because there is more tension in this spring, it's going to be open, but there's going to be some cases where the channel is going to be closed. But most often when it's bent in this form, it's going to be open. Over here, it is, I drew mine straight up and down, but this is actually in the opposite direction. This is going to be closed. Most of the time, if it's either straight up and down, or if it's towards the left, or I guess towards the more shallow side or the shallow gradient, it's going to be closed. It's going to favor the closed form, and but for some of them it is going to be opened. And so the other important point here too, that I haven't mentioned yet, is that when it bends, when the stereocilia bends towards the highest part, it's depolarized. And when it goes the other way, it is actually hyperpolarized. Meaning the charge is sustained within that cell, and it becomes even more positive. And so that's another thing to consider. Yes. Thanks for watching. So for this question, I'd like you all to pause the video and see if you can work this one out. The question is, how do the cellular transduction events differ in hearing from olfaction? How does this explain the speed differences? So hopefully you all have had a chance to work that out on your own. So I'm going to work this out really quickly. So on this side, I'm just going to do olfaction, and I'm going to compare it on this side with hearing. So in the case of olfaction, you have a odorant binds to the receptor, and then what you have is activation of G protein, and then the activation of adenylate cyclase, which then converts ATP into cyclic AMP, which then stimulates ion channels that depolarize the cell. For hearing, you have a mechanical stimulus that is generated from the moving of the cochlear fluid, the fluid in the cochlea, over the stereocilia. So you have the mechanical stimulus. Then what you have is this directly opens the channels, the ion channels, which then leads to depolarization polarization of the cell. So as you can see when you, so these are the differences. So how does this explain the speed differences? Well, in terms of signal transduction, you can tell that this olfaction has this secondary signal that it has to go through. So all of these steps right here are to activate the secondary signal that then goes on to activate the ion channels. In this case, you have a direct activation of the ion channels with a mechanical stimulus. This is called mechanotransduction, and it's far more direct and it's a lot faster. And so with that, you can achieve, you can detect time delays as low as 0.02 milliseconds with this system, which, to remind you, this allows us to be able, the quick processing allows us to be able to tell the direction that sound is coming from.